Thanks so much, everybody, and good afternoon. It's an honor to open up the session here. And I'm going to present to you a new project that I'm going to be working on in the foreseeable future. And as you can see from the title, it's about ancient DNA. And we'd like to be tracking Holocene genetic variability of Swiss mountain forests using ancient DNA. So let's see if that works here. There we go. So you all know that iconic figure from the last IPCC report. So climate is changing. It's getting warmer by the end of the century, about, depending on the models, 2 to 5 degrees rise in temperatures. So species are forced to also either move, of course, to stay in their climatic niche, or to adapt to those temperatures, or maybe in some extreme cases also go extinct. And in the Swiss Alps, as you can see them here in the background, species have it maybe comparatively easy. They can just, if they want to stay in their climatic niche, they can just move their distribution upwards to higher elevations where it's colder as well. But we also see, or we also expect a similar pattern on a continental scale. And here you can see, for example, output from a dynamic vegetation model, which is simulating distribution or foliar cover of silver fir, Abies alba, under 2 climate scenarios for Europe. And you can see, for example, here, under the high emission scenario, there is quite a decrease of silver fir in the southern range, and there's probably an increase in Central Europe as well. So there's a range shift that is projected here. And such range shifts also have a drastic impact on the gene pool of a species, especially if the rear edge might go extinct. As you can see here in this schematic graph, because the rear edge harbors a large amount of genetic diversity due to the older populations that might actually date back to refugial populations. So such an extinction of rear edge populations as they are projected here in the vegetation model output might have quite a drastic effect on the gene pool of a species. And lower genetic diversity, that might also be then a problem in adapting to changing environmental conditions. There are studies that show that high genetic diversity facilitate adaptation to changing environmental stressors. And so you all know here that, of course, if you want to know what the future brings, it might be worth to have a look at the past. So we all know that there have been already range shifts in response to past climatic changes, for example, glacial interglacial cycles. Here you can just see a schematic of a post-glacial recolonization of Europe of beech, of Fagus sylvatica. And you can see that, of course, beech survived the last glacial maximum in refugial populations in the southern peninsulas, so in the Balkans, in the Italian peninsula, and also in the Iberian Peninsula here. And then with warmer temperatures, could again expand into more northern latitudes here. And evidence comes from a variety of different sources. So from fossils, for example, here, those are the rectangles, and also from genetic information. And then what we can also use is we can use models to verify or to back project past ranges based on climate data and present-day species correlation with temperatures. So let's zoom into the Swiss Alps. That's where we are planning to work on a study here. Here you can see also the recolonization pathways based on the current knowledge that also, again, stems from fossil records and from phylogeography. And so what you can see here is that after the last glacial maximum, species kind of shifted the range northwards. Mostly it's a north-south shift, north shift. So from refugial populations in the south, they expanded into the Swiss Alps here. And we know all that, again, from fossil records and also from phylogeography. So we know that there, for example, this is the example of larch A study by Wagner et al. A couple of years ago showed that there are probably six different refugial populations based on present-day genetic variation. And combined with the fossil records, we can make such arrows there that represent possible expansion pathways into the Alps. And the only species that's a little bit different is Norway spruce here. Picea abies has a more east to west expansion, and that is also due to the refugial populations, basically. But the thing is, those arrows or those population expansion pathways, so far they only just represent hypotheses. So far there is no really a way to test those pathways, because fossil records don't give you a relationship. It just shows you presence, absence, and then of course you can infer directions as well. But there might have been other processes involved, such as long range dispersal and so on, or maybe populations that are popping up later on a later stage. And similarly, phylogeography only gives you like a snapshot of the present-day diversity, which you can, of course, use to infer past genetic variation. But there might have been populations that went extinct in the course of the Holocene, or, for example, if there's a human disturbance that causes a drastic reduction in population size, you might also lose a large chunk of the genetic information there. So what we propose in our upcoming project is to actually test those recolonization pathways using ancient DNA. So what we like to do is we like to sample 7 different lakes that are based on a northwest and on an east-west and north-south transect through the Swiss Alps, and then actually to check the relationship of the first recorded populations between those sites. Because we know from macrofossils when they appear, and then if we analyze the aDNA of such macrofossils, we might be able to test the relationship or those expansion pathways. This is important also to estimate rates of population expansion in regard to future climate change as well. And then maybe this could also be used to identify potential populations from cryptic refugia, so from populations that might have survived the last glacial maximum close to the ice sheets and are not detected with classical methods such as pollen analysis because of their small and fragmented population size. And also, of course, of populations, as I mentioned already before, that might actually just got extinct, that survived somewhere around the ice sheets in small fragmented populations, but then they got extinct or swamped out by populations that came from the classical refugia from further south. And another idea is that we don't just want to test those recolonization pathways, but we also are interested in changes in the genetic variability through time. So what we plan to do is we plan to compare the genetic variability of the past populations with the present populations around our study sites. So link back to present-day phylogeographic studies. And this would allow us to see if there are changes also in the genetic variability. It would also allow us to, if we specifically focus on adaptive alleles, to check for the adaptive potential of species in response to changing environmental conditions. And so how are we going to do that? We'd like to, of course, use again lake sediment archives, because we know they're a great archive. Because here we also have like a population inference from pollen and macrofossil data, and then we actually like to target specifically macrofossils of those 5 tree species that I've mentioned before. So from top to bottom, so Scots pine, Swiss stone pine, larch, silver fir, and Norway spruce. And we'd like to sample the first populations that establish at this time at this site, and we want to do that again, as I already showed, for 7 sites in Switzerland. And then from these macrofossils, we'd like to extract then ancient DNA. And for those of you that have been to the ADNA session, that might be now a little bit of a repetition, but for all the other that missed the ADNA session, I'd just like to give you a short overview of how we want to do that. We plan to apply a method that Nadia Alvarez just presented also in the ADNA session, and it just came out. It's based on a paper by Schmidt et al. Just came out on Monday. And just to give a quick summary, because it's very hard to get ancient DNA because it's very fragmented. And what we want to do is basically we use RNA probes from fresh specimens, so from modern RNA, and use those as baits to capture ancient DNA. So we're only interested in specific species. We're not interested in capturing like the entire vegetation that was present. So we're only interested in the genetic variability of those 5 species. And so if you have any questions about that, you can come to me afterwards. I'm not going to go into much detail. Or you can also ask Nadir about that. But the thing is, it's very cost efficient, and it also selects only for expressed genes. And it's not the entire genome, because we're only interested in single nucleotide polymorphisms of a reduced genome, because we don't want to have the entire picture. We just want to have a statistical a sample of or a random sample of the genome, so we can do our SNP analysis. And just to show you as well that it works, again, those of you that have been into the ADNA session might recognize this figure here. So just to quickly summarize it, here you have silver fir from a lake in Ticino. You see here, this is on a time axis here. So you have, this is just a schematic of the range of silver fir, basically. Then you have a disturbance here caused by human impact. So the first farmers in that time settled there, burnt down the forest, which led to a fragmentation of the silver fir population. And then you can see a subsequent re-expansion of silver fir in the area before it then, in the Bronze Age here, completely disappeared in the region. And so there's a few very interesting ecological questions we can ask. For example, is the re-expansion caused by trees that come from outside and repopulate the area, or is it caused by the surviving trees or surviving species or surviving individuals around Lake Aurelio here? And Schmidt et al., they sampled ADNA from, again, macrofossils from those needles at different time slices here, and we're actually able to show that there's so here you see the genetic variability, basically, so it's observed and expressed heterozygosity, and you can see there is a decrease in genetic variability during the decline, but after decline the values go back up to where they've been previously, so prior to the disturbance, which indicates that this re-expansion was caused actually by the trees that were present at the site, so not by individuals from outside. So just to sum up again, we think what ADNA is really an amazing tool for. We think we can test recolonization pathways of tree species, and we might also be able to maybe possibly identify cryptic populations that do not show up in pollen analyses. And what we're really interested in is to study such population genetic processes over long time scales. So we're interested in, for example, founder events, genetic drift, or gene flow that we might be able to pick up with ADNA analysis. And another question we're interested in is to estimate the adaptive genetic variation of tree species in response to climatic changes or just environmental changes. And the overarching goal actually of this project is to assess the impacts of global change on mountain forests on a variety of scales, from the landscape scale to also the genetic scale. Okay, so thanks for your attention, and I'm be happy to take questions if I have time.
Could you say something about the preservation? How many needles do you have to look at to get a success?
Well, of course, the more the better. You know, that's always the case. So the study of Nadir, you can see actually the sample size here, but that was only a preliminary study. And we'd like to increase our sample size to about 40 needles. They found that about 60% actually have aDNA in it that can be processed and used for the genetic analyses. So that would give us like 20, 30 samples, which is of course still just barely enough to do any good population genomic analyses. But yeah, again, the more the better, and we just try to scrape together as many needles as we can in as short a time slice as possible.
I saw the title of this session, which says that if species persist, survive, die, or adapt, which is really, I think the topic of this talk would fit it nicely. This work has been carried out with my colleagues, all these colleagues, in the frame of this European project, EcoChange. And we looked at the temperature, the range of January temperature for 3 species, silver fir, spruce, and beech, over the last 10,000 years. So I was saying that the idea of species, do they adapt, move, or die? In fact, when we look at this data, we can have some clues about these 3 questions. In terms of moving, and this is something that is now more or less clear. This is the IPCC synthesis, and these are some data that we have about the migration rate of silver fir in Europe. And the data that we have fit more or less nicely all the data that we have in Europe. In other words, the data that we have for the Holocene, the migration rates of many plant species are much lower than the rates that are needed to track the 21st century. When I say much lower, it's about 10 times lower. These species, silver fir, migrates by about 1 to 2 kilometers per decade. And the expected rate of climate change is about 10 times, something like 10 kilometers per decade. So can they move? Well, actually not very much, because these are the trees, and this is the IPCC synthesis with different climate scenarios. I'm sorry, you can't see it very well, but on the right-hand side, you have different RCP scenarios, and the last one here is the RCP 2.6, and the summary of these RCP scenarios are here. The last one, the lowest scenario, 2.6, means that we should remain below 400 ppmv for the next century. In that case, and in flat areas, trees can move or can persist or can cope with the climate change. Otherwise, for all the other climate scenarios, they cannot be compared to monkeys or butterflies. So we have to find some solution to preserve them. There are a few solutions. The first one is to keep them in situ, of course, or translocate some seeds to suitable habitats, or create some corridors with some stepping stones populations for their potential migration with the help of human beings. But in any case, whatever the decision that we will take, we will need some modeling tools to predict where will be these suitable areas. This is something we need. We can choose any solution. We need to predict the areas where the species will survive. This is a very old concept made by Hutchinson in the late 50s, which says basically that each species, animal or plant or whatever, any organism has a niche. And this niche, the niche of this organism is defined by different parameters, like precipitation and temperature in this case. And in this concept, of course, there is the fundamental niche, which represents the largest or the widest range of all the conditions that constrain the species in the niche, in the geographical niche, and without competition. This is very important. And the realized niche, which is just a part of the fundamental niche, which is which corresponds to the areas that are occupied by the species when competition is taken into account. So this concept has been used by ecologists for many years until late 2000, the last 15 years. And then came the computer era and computer time and statistics and so on. And this concept has been used to predict the species using the areas occupied by species and the climate where they are, using statistical methods and using that calibration to predict the species' future areas. So this concept, these new tools have been used for many purposes, and this is one of them, made by Jan Svinning and Skov. And in fact, they used a niche-based model to predict the potential area of the three species on which we are working, silver fir, spruce, and beech, compared to their realized niche. The realized niche is in black, and the gray one is the potential one, that is the expected area where the species should be. In fact, there are many species, according to this study, they do not fill their entire range today. And in the case of silver fir, today it occupies about 50% of its potential range. So this is one use of this niche concept, and it shows, of course, that the dispersal is a major variable that controls the realized niche by this species. In fact, when we look at this type of modeling, so far it has been taken only the modern distribution and the modern climate, making a calibration and making the predictions. But there are very few studies where it takes into account the dynamics change of the niche itself. I like this sketch here made by Peter Perman and his colleagues, where they show that they made a theoretical sketch to show that, in fact, the fundamental niche and the realized one at a certain time t can change when the species adapts or do something. So the red one is the realized niche that switches into fundamental niche, and then it moves on, and then we end up with a new realized and new fundamental niche through time by adaptation process. So I talked about the moving species, not very possible for trees. Adaptation is definitely possible when we take into account thousands of years. Of course, for the next century, we are talking about few decades. We are not talking about years. So adapting is a nice thing if we're talking about geological times, but not when we're dealing with 1 century or less than that. In order to take into account this species adaptation through time, Luigi Marano and colleagues used this idea, and instead of calibrating the model only on the modern distribution of the species and the modern climate, they have used simulated climate between 13,000 and today, and the species occurrences between 13,000 and today. The occurrences were taken from the European pollen database. So for each time slice, for 13,000, we have the occurrences from pollen, and we have simulated climate. We calibrate the model, and we do that for each time slice. So in fact, or somehow, I would say, Luigi took into account part of this adaptation process of the range of species. It's not an adaptation in terms of genetic adaptation. It's just a geographical adaptation of the species. So using this calibration through time between 13,000 and today, Luigi Marano and his colleagues made these predictions for these 3 species, for silver fir, spruce, and beech. of course, this is a projection for the next decades, for the next 50 or 80 years to come. So no one can tell today if it is going. Sorry, I'm switching from one language to another. If it's going to be if it's realistic, more realistic than just making a collaboration between the modern species and its climate, or better. We don't know. So this is something. This is the state of the art so far. Looking at this work, which has been based on simulated climate, we thought, with the same people in that work, that this work has been done also in the EcoChange European project, we thought that it would be a good idea to look at this type of relationship between species and climate, but using only data, not using any simulations, neither for the species distribution in the past, nor for climate from climate models. And this is how we came with this idea that we should have reconstructed climate, quantified, and species occurrences from the past, and try to see how the species reacts to climate. So we reconstructed climate, and we used, this is a very basic slide, which shows that, in fact, we have used a kind of mutual climatic range, which is somehow the species climatic distribution in its space. And for each fossil sample, we use the taxa that are in the fossil sample to get their climate separately, and then we combine the climate information for each species in the fossil sample to obtain a common or joint climate. Okay, so we have used this method to reconstruct climate for Europe. And this is what we have obtained. So we have a database of several tens, between 200 and 300 sites. It's the dark points. And for each dot, we have one climate value. And for each time slice, 10, 9K, 8, 7, and so on, we have interpolated that value over the space. In this case, I have used the universal Kriging, which takes into, it's a kind of cube. You take latitude, longitude, and altitude, and projected the reconstructed January temperature over Europe. I'm going to try to be fast. So this is climate. Of course, in order to avoid a kind of circularity in the reasoning, we did this test. In fact, we removed one species, silver fir, we removed beech, we removed picea, and we removed the three of them from the climate reconstruction. And we compared, this is for the modern, we compared the reconstructed modern climate from pollen by removing all these three species and compared it to the world clim data. And the deviation is less than 1 degree. So the reconstructed climate that we have reconstructed for the past may deviate by, say, 1 degree, but it's robust. For the species occurrences, we have used pollen and macrofossil remains from the literature. So here we have climate that is more or less robust, and species distribution that is more or less robust too. The last slide. Okay, so we ended up with this situation. This is today the distribution of spruce, beech, and silver fir. Orange is the distribution to where the species dominates, and yellow is the part where the species occurs. Say this is okay. The orange part is between 25 and 75% of the climate distribution of the species. To sum up, in fact, we see that there are major shifts in the species distribution. And just to talk about one species, so I won't take it that long. For instance, for spruce, the dominant part of the species can stand today up to 4.5 degrees in terms of January temperature. But its edges, mostly the warmer edge, cannot stand more than 1 degree. So the reaction of the main parts of the species and its edges will not have the same reaction to future climate warming. I'm not going to go through this because it will take too much time and I don't have time. So there are some conclusions, some cautions here. the conclusions is that, of course, the fossil data can provide some valuable information about tolerances and can help for predicting, but also at looking at these edge versus dominant areas and so on can be useful. They can also, the temperature changes may have affected different parts of the range, not only its main part. But there are also major cautions in this type of study. Of course, all climate data need to be constrained by either other proxies or use other proxies. In any case, this is just a piece of work that needs to be improved. And also, one climate variable is, of course, does not explain the species niche. Of course, we need at least two or other or more climate variables. Thank you so much. Muchas gracias.
Good afternoon, and thank you very much. The talk I will present today, turn around the Aves alba, the silver fir. We will discuss first for the problematic at the Pyrenees level, and second, the project will be described, and at the end, the fir results will be shown. Well, let's discuss why we are interested in Aves alba. Taking a look on its distribution, we can see that in the past it was much wider. On this pollen curve, we can see that it's a common phenomenon in several European regions, and even we can see an important past presence in areas where today is completely missing, like here in this pre-Pyrenees area. Well, the current situation has clear threats too. First, we can speak about the potential distribution area is larger than the current distribution area according to the topoclimatic models of suitability. One example of the suitability models is present in the European Atlas of Forestry Species. At left, the current situation, and at right, the suitability model. The difference is quite clear. And second and third, the actual area is fragmented and confined generally in the northern slope. This map shows quite well this fragmentation, and with this small graphic, we can see this predominant north orientation. Well, and the future is full of unknowns and questions. Some studies suggest the reduction in response to the expected warm, and others suggest the stable conditions or the expansion. Well, under this problem, the project that we present today has an objective: explain the current distribution at its ancient evolution with the aim to provide background to anticipate the future development. We are interested in all topographic and altitudinal distribution, as well as the ancient refuges and the migration paths after the glacial period in this Pyrenees area. Sorry. To answer all these questions, we have to work with extended time scale, the Holocene period. It's quite clear in this period both natural aspects and human activity participate in the forest development. Next, we address this work with a paleobotanic interdisciplinary approach. And finally, high spatial precision is an important aspect in our research. We are interested in what happened in a valley, but also in which part of this valley, in which slope, or in which parcel. So the main methodology used is based on the soil charcoal or the paleoanthropology. This discipline used the charcoal with the aim to reconstruct past woody vegetation. It allows us to work at parcel or slope level. It is possible to quantify the charcoal in the soil, to identify its species, and finally, we can date these charcoal fragments. With all this, we do the interpretation. Well, the pictures show the process, the field work and the charcoal isolation, And finally, the taxonomic identification of the charcoal. We have planned 5 sampling areas through the annex from coast area to the Pyrenees. In each area, it has been planned topographic transects of sampling. Thus, we work with 2 special scales, local and regional. Well, the pollen data of Central Pyrenees show the phylogenetic advance from east to west. Here. And the disturbance periods like early Neolithic, Bronze Age, Roman period, or medieval period. And the successful period, with high presence of fir around 4,000, 5,000 CalVP. In Bur Lake, we can see clearly this last glorious past period with pollen and macro remains. Bur Lake, like you can see here, is located in this southern slope. Thus, we can assume that the Abies was present in this south-facing area before Bronze Age. Well, today is completely confined in the north area. Well, the first pedantropological area is located in Andorra, in the axial Pyrenees, around 15-17 meters above sea level. In this area, we have some fir in the northern slope, and Pinus ancinata and Pinus sylvester in the rest of the valley, and some little young firs in this west area, in this western slope. We have sampled here the north area inside the fir woods and southern and western slope. Well, here in the center you can see the soil profiles, and at right the results of species identification. In green you can see the Abies charcoal fragments. Well, the first thing that we can see is the past presence of Abies in all the sampling points, north, south, and west. So it confirms the past fir presence in the areas where today is missing. Well, it's clear that the south-facing signal is weak, but it demonstrates that Abies past presence in a recent period, around medieval and modern periods. Early Holocene fir past presence is registered in these western areas. Another aspect to be highlighted here is the important dynamic interactions within the species in all the points. There isn't a monospecific sample point. And last, just point that we have detected the ancient presence of Taxus baccata, another species today missing in the area. So, the conclusions, we can assume the methodological complementarity of soil records and sedimentary records. After the confirmation of a past wider distribution of Abies during the greater part of the Holocene. Next, the early presences in sunny areas at the same time as a recent presence around medieval and modern periods. Also, the Abies alba range distribution has been determined by the interactions among other 3 species. And also the fire like an important factor of forest dynamics in the Pyrenees. And finally, the present-day recolonization of fir tree that involve an important forest dynamics for all the exposures. And thank you very much for your attention.
Can I ask you about the preservation of the charcoal and the identification of the Abies charcoal? Is that quite common that you can nail that down to that species? Or?
The conservation is quite good. The difficult, perhaps here, is the small tail, the small size of the the small size of the charcoal. Okay. The problem is that because we work with charcoal around 1 millimeter or less, so the difficulty is here. But the preservation is good.
And I suppose the taphonomy of the charcoal, when it gets that small, is quite difficult as well. I'm imagining the distribution.
Sorry?
The taphonomy, the distribution of the small charcoal shards.
The distribution in the soil.
Yes.
The problem here is in the soil, there isn't a chronological stratification like sedimentary records. So the difficulty is here also. We have the high precision because it's in the area. The information it's very local, but it's true that in the chronological information it's quite weak. So we work with the registers and the sedimentary registers and the soil registers.
Okay, good afternoon, everyone, and thank you for this opportunity to talk about our research. Paleoecological studies are more important than ever because they can address issues of current concern around natural population dynamics prior to human impact, the exposure of species to climate change, and their vulnerability and resilience. In particular, trees are very interesting in this respect because they're long-lived organisms for which modern ecological observations may only represent a fraction of their lifetimes. And of course, there's a current imperative in the face of global change to use this type of information to inform conservation and management strategies. The focus of my talk today is the Atlas or Atlantic cedar, Cedrus atlantica. It's an endemic conifer to North Africa. It's distributed, shown in green on the right there, in a fragmented distribution across the mountains of Morocco and Algeria. It's concentrated in an elevation range between around 1,400 and 2,600 meters above sea level. Despite that fragmented range, around 80% of the forested area is concentrated in the Middle Atlas. That's number 3 there identified on the map, and that's the focal area for our study. The Atlas cedar is currently considered an endangered species on the IUCN Red List, showing signs of unusual mortality across around 75% of its range. And multiple drivers are implicated in these changes, both climatic, anthropogenic, and biotic, but a key emphasis is on the important issue of drought impacts and temperature-driven drought. In this respect, it represents an important global case study for the theme of forest dieback. And ultimately, the fate looks very bleak, as Rachid, who spoke two talks ago, has highlighted that it's ultimately predicted to become extinct in many of its core areas, possibly within the next century. What can we hope to add from further Holocene palynology? Well, there's a very interesting question around the process of colonization, how this particular species has moved and its range has changed in the past. The Middle Atlas that today is the core area was not an important area for Cedrus atlantica in the early Holocene. Along the way, with high-resolution records, can we document the response of this species to past climate changes? And ultimately, can we add something to the understanding of its exposure to climate change and its resilience in the face of past abrupt changes? The Middle Atlas is a wonderful place to work in terms of paleoecology. There is a diverse range of wetlands, from permanent lakes through bogs, all set within this karstic landscape. Bringing together the available records for Cedrus atlantica, the diagram here shows curves from previous studies. The map shows the western area of the Middle Atlas where paleoecological studies have been undertaken. And overall, the data that you see here highlights the mid to late Holocene colonization and expansion of this tree in the Middle Atlas. The timescale there is 12,000 years, as it will be throughout the talk. But looking at the evidence to date, we also see that there are relatively few records that extend into the early Holocene to illustrate the process of colonization, and also there are relatively few records on centennial timescales, which become increasingly relevant for rapid change. The site that I'll present data from then is Lake Sidi Ali. It's a lake located at high elevation, above 2,000 meters. The lake's origin is mixed in terms of structural and karstic processes. It's a closed system with no major fluvial input. The lake has a large surface area. It's one of the largest lakes in Morocco, almost 3 square kilometers, but variable with precipitation. The catchment area in terms of overland flow is very small, and the maximum water depth is around 35 meters. These particular characteristics, from a palynological point of view, mean that the site is an ideal receptor for regional atmospheric deposition, getting an integrated signal of changes in the wider region. And ultimately, the site is also of great interest because of its sensitivity to changes in hydrological balance. And these features of interest were, of course, what promoted and drove the pioneering work there in the late 80s and 90s by Henry Lamb, Phil Barker, Neil Roberts, and others, many of whom are here in the conference this week as well. Our approach is a multi-proxy one, incorporating physical and biological and geochemical insights into the lake system, but what I'll focus on here today are the terrestrial proxies that we've mainly worked on in Manchester, including the pollen, microcharcoal, and then underpinned by a timescale that's built on lead-210 and 137 cesium constraints and 26 AMS radiocarbon dates, primarily on pollen concentrates, terrestrial pollen concentrates, to avoid hard water error. So here's the pollen record from Sidi Ali, again spanning the last 12,000 years, offering subcentennial scale resolution. And the overall picture that we gain from the pollen record here is one of a series of gradual changes in the predominant biome around the site, such that in the very final centuries of the Younger Dryas and into the earliest millennia of the early Holocene, we see dominance of a steppic landscape with Artemisia, with Pistacia, and with Lygeum spartum, a characteristic steppe grass. Moving into the early to mid-Holocene then, we have the dominance particularly of evergreen oak, shown in the olive color there, accompanied by Filaria, along with grasses, picture of an open sclerophyll woodland predominating. Into the mid to late Holocene then, we have the dominance of cedar, along with junipers, and some representation of deciduous oak, pictures of a more montane-type forest. And yet, during that interval, slight indications also of development of scrub taxa. We see Ulex type in pink there, and also, as evidenced by the charcoal accumulation rates, higher fire activity. Finally, in the upper part of the diagram, then we see indications of human impact with a decline in the total forest cover affecting most species in a pretty even way. So I'll focus on the curve for cedar here as an indication of the fluctuations in cedar population through the Holocene. So in terms of the kind of climatic drivers that may be important, overall we see a sort of late temperate pattern that may well be linked to long-term tracking of orbital scale climate changes associated with the precession cycle. A key factor may be reduction in the seasonal extremes, cooler summers as we move into the mid to late Holocene, associated then with greater availability of growing season moisture to this particular species. We also see, if we look at the millennial scale then, this curve shows sea surface temperature changes off Cap Blanc, off West Africa. Famous study from 2000, Peter de Mennecal, where also the end of the African humid period was identified. And what we see is a very nice match between millennial scale cooling events associated and linked. At the time, the great insight from this was the connection between these cooling events and the high latitude bond events. And what we see is a positive response of this particular species on millennial timescales to the Holocene cooling events, associated tentatively with cooler summers, again, enhanced growing season moisture, and possibly enhanced precipitation coming from the Mediterranean, a shift in seasonality of moisture perhaps as well. It's also interesting to consider the biogeographical implications of the curve and its tendencies. Worth pointing out that the main features are supported by the concentration curve and the accumulation curve as well. In the early Holocene, up until around 6,000 years ago, we can consider a migration or colonization period, and the kind of dynamics that we can infer to be taking place, long-distance dispersal events from areas where cedar may have been more abundant prior to this in other mountain ranges of North Africa, an aspect of leading-edge dynamics as this particular tree attempts to colonize and then experiences episodic extirpation, and also periods of slow recovery between the few millennial scale expansion events. As we move to the mid to late Holocene, then, we can consider a tolerance phase characterized by stable or more stable in situ populations. We can envisage an aspect of trailing edge dynamics as cedar competes with other species, notably junipers. There are indeed episodic declines, but not leading to total extirpation, and generally insights into rapid jumps that may be associated with rapid recovery or expansion of the populations. And these little arrows and red bars are to highlight a slightly different structure in the two parts of the record, so that in the early Holocene we see sort of slow recovery leading to a temporal maximum in cedar followed by extirpation events, while in the mid to late Holocene then we see the tendency is for gradual decline culminating in episodic declines and then followed typically, for example, there in episodes of rapid recovery. Now, my question as I started to look at this was, does this represent a sort of one-to-one tracking of climate on centennial timescales? And to explore this a little bit, I have created a very simple model that attempts effectively to simulate the pollen curve. So this is the cedar curve that we've seen already, turned on its side. And what I've done is created effectively a climatic or an environmental signal that incorporates aspects of what we understand to be typical of climate on the long run. In other words, components fluctuating in an effectively sinusoidal way on different timescales. The timescale isn't really realistic, nor are the scales of the population, but it incorporates elements of different frequencies that could represent, for example, the orbital scale, multimillennial, millennial, and centennial scale variability. Now, inspired by a publication by Stephen Jackson, the idea here was then to see what happens if you set a simple threshold and allow the population to expand when it exceeds that threshold. And I've run the experiment in two ways. The upper part relates to the two red lines. This incorporates a high threshold for recruitment and a relatively low threshold for survival. The green line represents a single threshold. Above that threshold, cedar expands, below it, it declines. And if we compare the two results, we can see that of the two models, A incorporating two thresholds, we can suggest that this is a slightly more realistic simulation. It captures aspects of that pollen curve in a better way, where we include separate thresholds for survival and recruitment. The second appears unrealistic. With a single threshold, we get a very spiky, dynamic response where we see effectively every change in the fluctuating environment recorded. If there is some element, I appreciate that it's a very simplistic type of setup, but in this way, some of the messages that I think are important are that the peaks that we see in the cedar population might correspond to very brief windows of recruitment. And those windows of recruitment might be separated by quite long intervals where the population is surviving but not necessarily expanding. This type of shape in A, well, you can debate with me whether you think it looks at all like what we see in the Holocene record, but it also depends on having a sort of fulcrum somewhere in the onset of the late Holocene, after which conditions become less favorable. And part of this is that we see then aging populations with low recruitment for more than millennial intervals. So to conclude, we see a Holocene history of colonization, establishment, and decline. We see that there may be climatic drivers for the long-term and millennial population size. And we can also consider in biogeographical terms a shift from colonization to tolerance. And from the modeling, I think we may anticipate that there may have been relatively brief windows for recruitment and regeneration. So just to answer that final question, are there Holocene analogues for current dieback? Well, there certainly are recurrent episodes of contraction. And some of the key insights from this record is that the species demonstrates strong exposure to Holocene climate changes, but that perhaps some disequilibrium with climate on centennial timescales may not be unusual. So leaping ahead then to the final point here is that conservation efforts should probably focus on preservation of mature specimens that can survive for long periods, as well as translocation schemes. I'll stop there. Thank you.
more sites to define the area, or were they in more humid places?
It's a very good question, and I think it's one that you struggle to answer from a single record. I think the defining characteristic here is that the Lake Basin receives a very large pollen recruitment area, which makes that a particularly pertinent question. I think it's only one that's going to be answered in the long run with additional sites covering this period of time. Hopefully in the future also insights from macrofossils as well. As I know, Rachid, you mentioned that you're on the hunt for potential micro-refugia, for example, something very important to understanding these type of responses we see to climate change.
I think it's also interesting, to study the timberline dynamics there, because I think it's almost unknown.
Absolutely. It's something we see when we start to put together the records from multiple sites, is an idea of those elevation changes that are implicit here as well. Yeah.
I would like to thank you, first of all, for the opportunity to give this talk. My title is a little bit different, but the content is the same. So today I'm going to be talking about reconstructing forest composition from fossil pollen data using some sort of new statistical methods. And so this work is part of the PALEON project, which stands for Paleo Ecological Observatory Network, which some of you in here are part of, and some of you have heard of, and some of you may have never heard of. So we know that trees and forests respond to climate, and a lot of work has tried to understand how these forests are going to change in the future. So we know things are going to change, things like composition, richness, and other forest characteristics, but we don't really have a good idea how. And so one of the ways that we quantify forest change is using ecosystem models. So there are many ecosystem models, and there is not great agreement among model predictions of these ecosystem variables. So we can't get it right for the past. We can't get it right for the present. So what hope do we have of getting it right for the future? That's not supposed to be it. Oh, sorry, guys. So one of the things that we focus on with PALEON is trying to improve these ecosystem models using paleo data. So ecosystem models can predict vegetation on the landscape at any point in time, including the past. And at that time, we have this paleo-ecological data that we don't really make use of in these ecosystem models. So we'd like to constrain them. But we can't plug in fossil pollen into ecosystem models. So we need to do some sort of modeling to translate the pollen or any other paleoecological data into something we can use. And so when I'm talking about paleoecological data, with respect to the part of the project that I work on, I mean fossil pollen data. You guys probably all know what that is. We've seen lots of talks about it today. So trees make pollen dispersed in the air. Much of it ends up in the sediment through a series of complex processes that include differential production, dispersal, sedimentation, among many other processes. And so what we can do is we can try to translate this pollen into vegetation. So pollen is not quite the same thing as vegetation because of those complex processes. So people have been thinking about how to do this for a really long time. Just last year, we celebrated the centennial celebration of the pollen diagram. And that was a celebration of Leonard von Post. And a long time ago, in his field notebook, his motto was think horizontally, work vertically. So already that far back in time, he's starting to think about how we can use pollen records to understand changes in space through time on the landscape. So today I'm going to talk about what we call steps. And so this is a pollen vegetation model that builds on theory that many others have developed before us. And so in order to do this pollen vegetation model, we need to somehow be able to build a model that links pollen to vegetation. So we need a point in time for which we have both data types. So that is now. But we know that the pollen-vegetation relationship depends on forest structure and other forest characteristics. And there has been research that shows that the pollen-vegetation relationship has changed as humans have modified the landscape. And so what we can do is For some parts of the US, we have this cool, it's called land survey data, public land survey data. So prior to Euro-American settlement, surveyors went out on the landscape and mapped forest composition on a grid. It wasn't meant to be an ecological data set, but it provides us now with a snapshot of forest composition in time. And so for this model, we're really interested in thinking about regional scale changes. And so in order to do that with some certainty, we need lots of data. So we can look to the paleoecology database. We have one that's in North America. Sorry, I'm a little nervous. Lots of people here. So we can look to the Neotoma paleoecology database, where there's tons of pollen records. And so our group contributes to this database, but us alone, we don't have the power to go out and collect the data we need to do these kind of large-scale reconstructions. And so we start with the upper Midwest. So people have been doing paleoecology here for a long time, so there's a wealth of paleo data available there. So this is a distribution of the fossil pollen records on the Neotoma paleoecology database for the upper Midwest. So there's a lot. And we also have this public land survey data, so the snapshot of forest composition in time that we can use to calibrate, to build this pollen vegetation calibration model. And so we can make these maps for this calibration time of our sediment pollen data, so our fossil pollen data, at this pre-settlement era, and our survey data, our public land survey data. And so we can kind of flip back and forth. Let's see if I can do this. There you go. And so it's a little bit hard to see here, but the more similar these are, the easier it will be to build a model. But we know that they're not one-to-one. So the question is, can we build a model to account for the processes that make these dissimilar? And so this model, the acronym is STEPS, which stands for what's underneath there. I'm not going to say it right now. And it's like many of so there are other models that are similar, so have some aspects that are similar to STEPS. So thinking about local versus non-local contribution, differential production, dispersal, things like that. And so I already mentioned this. We have a time for which we calibrate the pollen-vegetation relationship, so over there on the right. We have that land survey data, pollen data, and then for the times for which we only have pollen data, we can go back and estimate that latent vegetation composition. And so I'm not going to talk about the details here, but the model sort of looks like this. So we have some pollen counts that we can model directly, multinomially, as a function of the effective contributing pollen, scaled according to a differential production parameter that we estimate. The contributing pollen, or effective contributing vegetation, is determined as a sum of a local and a non-local component that depends on a dispersal kernel. And so there are parameters in all of these places that we can estimate all at once. We don't need to plug anything in. And so in order to go ahead with prediction, it's important to assess whether your model actually works. So when we calibrate, what do we see? How good are our results? And so I'm not going to talk about this in detail, but the red crosses are pollen versus vegetation. So if we just assumed that we could predict vegetation from pollen, so it's not one-to-one. Black dots, that's like a model versus data. So we're more along the one-to-one line, which is good. We're not perfect. And so if you're interested in reading more about these methods, I encourage you to check out this paper in QSR that we published last year. And so these methods are a step towards understanding. And some of the advantages to using methods like this are that we can exploit the spatio-temporal dependence in the data. So pollen data has dependence in time and in space. So vegetation, we can make some assumptions about how quickly things change across space. We also have this sort of cohesive hierarchical framework that allows us to estimate these process parameters without having to plug things in. So we don't need to know anything about pollen productivity or anything about dispersal when we build this model. Finally, we can explicitly account for uncertainty. Uncertainty in the measurements, uncertainty in age depth modeling, which I didn't talk about here, and uncertainty in the processes. So one of the cool things we can do, because we have this uncertainty, is make these maps of significance. And so we're thinking about significant change in composition through time. And so we can determine significance by comparing points of time, so pairs of times, and then we can make these maps and try to see where things are increasing and decreasing. So the details here aren't so important, but we can learn about where in the range something is increasing, where it's decreasing, and what time period that is happening in. We can also start to do some investigation to see how things are moving across the landscape. And this isn't really a range expansion because it's hard to identify the edge of a range. But we can see here we have some increasing hemlock through time and then a crash at the same time as settlement. We can also start to think about ecotone shifts because we have a spatio-temporal model. So here we actually don't see much shift in this transitional region, but you could. This is one thing you can do with this method. And then we can ask why it's shifting. So here it's because pine is increasing along that ecotone. We can also start to think about the emergence of novel communities. So for example, in the upper Midwest, we have the emergence of the Minnesota Big Woods. So we can identify which taxa are contributing to that emergence of this novel community and when it's happening. So finally, I just want to sum up. So today I gave you guys a quick presentation of this new pollen vegetation model. So we use this to predict past forest composition with uncertainty. One of the things that we do that's a little bit different, because we have this wealth of data, is we can calibrate this model prior to major land use change. And so kind of the big conclusion of this work is that prehistoric forests are not in steady state, unlike what many ecosystem modelers kind of assume. They might not believe it, but they make that assumption. So the next steps here are to go further back in time. So here I just presented results for the last 2,000 years before present. We'd like to go back as far as possible, making some big assumptions about the pollen-vegetation relationship. We'd also like to compare to other methods like love reveals, which works quite well in many cases. And so it's similar in many ways, but different in some other ways. Finally, we want to improve efficiency and make this more accessible. And so I'd be happy to talk with anyone after. Thanks.
The conclusion that forests are not in a steady state, this is an interesting, you may comment this a little bit more, what you have found, because this is very interesting.
Yeah, it is interesting. I think the first question is what do you define as steady state? Sorry, that's my timer. So how do you define a steady state? And different people have different interpretations of what it means to be in steady state. And so, I mean, that's going to determine what you conclude. But I would say things are changing. They're not huge changes. You know, if we go further back in time, we would see much bigger changes. But they're not. You know, there are some significant changes.
Good afternoon. Today I'm going to present this presentation titled "The Making of the Forest: Past Human Impact on Species Distribution in the Southern Brazilian Highlands." This is part of a larger project that is called Gel Landscape of Southern Brazil, integrating archaeology and paleoecology, that is funded through the Arts and Humanities Research Council of the UK and FAPESP from Sao Paulo. Well, we are concerned with the expansion of Paraná pine or Araucaria angustifolia mixed forests in southeastern Brazil. These forests once occupy 250,000 square kilometers from the southern section of Minas Gerais state to the southern part of Rio Grande do Sul, above 600 meters above sea levels. These are trees that usually get to 40 meters in height, and whose usually the diameter at the breast height is 1 meter. Well, we are very interested, obviously, in the role of humans, and we know that these Paraná pine nuts obviously were a major element of the native southern year groups' diet that inhabited the area from at least 2,000 years before the present. These trees are very productive. Each tree can produce up to 30 cones containing 112 seeds, are an excellent source of starch, dietary fibers, and minerals. It can be eaten raw, roasted, or made into a paste. It can be stored in tightly closed soaked basket, and also indirectly, when the Paraná pines are ripe, they attract a lot of game that is obviously very good for animal proteins. It doesn't come as a surprise that in the ethnohistorical period, these groups have a tight control over these forests. They were demarcated, and there were a reason to go to war. And also, most of these group gatherings, the Southern Jay, occur in autumn when the Paraná pine nuts are ripe, and also the maize is ripe. I don't have time to go into the cosmology and the symbolic part of Araucaria, but it obviously plays also a major role in all the from cosmogony myth to most of the rituals of the Southern Jay people. Anyway, 10 years ago, I'm going too far back in the past. 10 years ago, with Herman Behling, we take a look at the 10 existing pollen records in the southern Brazilian highlands, and we notice that around 4,000 years before the present, there is an initial expansion of these forests, but more interestingly, just 1,400, 1,000 years before the present, we see this massive expansion of Araucaria forest, from which you only have a sample here. Okay, so all these areas of campus were transformed into Araucaria forest. One of the more striking records is Cambará do Sur. Here we have the pollen record, where starting 4,000 years before the present, but mainly 1,000 years before the present, we see the complete replacements of high altitude grasslands called campos by Araucaria forest. This has been replicated by our own studies in the region. This is a work in progress by Macarena Cárdenas, where we see the same pattern of initial expansion of the Araucaria forest 4,000 years before the present, and a major peak 1,000 years before the present. This expansion of the Araucaria forest takes place in a period that we archaeologists call like hot periods, transformational periods, and this 1,000 years ago is when we see a major increase in southern Jay sites. But also very interesting is 1,000 years ago we see the arrival of monumental architecture. We see the appearance of mountain enclosure complexes, and also we see the appearances of what we call oversized pit houses. Most of these subterranean houses, habitation sites, are about 10 meters and up to 2 meters in depth. Well, at this time we see large communal houses that can be more than 20 meters and up to 5 meters in depth, coinciding with the expansion of Araucaria forest. Well, we are archaeologists, so the obvious question for us is, was this forest expansion driven largely by a change to wetter climatic conditions, or did land use by these proto-Jay groups play a significant role in the expansion of these forests, and if so, how? Well, one of the problems that we have with the extant paleoecological data, as you can see here, is that most of the archaeological sites, many of them are in the valley bottoms. This is Uribici in southern Santa Catarina, and most of the pollen records, like the pollen record studied by Herman Behling, Morro da Igreja, is very far away from the archaeology. So one way that we are mitigating this problem is by taking course very close to archaeological sites, and this is something that archaeologists interested in the role of climates, but also humans. We have been doing it for a while, trying to integrate what we call on-site, the archaeological site, with off-site paleoecological records, something that I have been doing from the early days of my dissertation in southeastern Uruguay. Here we have a circular village, and here we have the pit bog. And this is something that we are doing with Francis Mail now for almost 10 years in Amazonia, combining archaeology with paleoecology. Anyway, looking at the topic of this symposium and trying to model the distribution of Araucaria, we are pretty much based on the seminal work of Brazilian biologist João Rodrigues de Matos, who classified the forest in black forest, the forest that is very close to streams, Araucaria forests that appear in patches within campos, within grasslands, and some isolated trees. The more important thing that we saw from this work is what he says, that when Araucaria occurs in patches in the grasslands, they always develop in the southern slope of the hills due to greater humidity and better conditions. It never advances to the top of the hills or to the north-facing slopes, except when brought by external vectors, mainly humans. And here we can see some examples. The south face completely covered by Araucaria forest, where the north face is covered by campos. And here we have another satellite image showing the same. Well, based on these parameters, taking into account the aspect and the topographical position of the forest, we went to areas where there was no archaeology, and we modeled the maximum extent of this natural distribution of Araucaria forest. Here in pink, you can see our model, and as you can see, it predicted very well. In areas where there is no archaeology, we can see that the Araucaria forest is pretty much restricted to the south-facing slopes, with no forest in the northern slopes or in the plateaus. We are seeing a very different picture in the area where we have been doing archaeology now for 40 years. We have documented more than 50 sites. We have dug 11 of them, and here we are seeing that we see Araucaria forest in the top of the plateaus, and we see Araucaria forest beyond the south-facing slopes. So this is the area of Campobello do Sul, where we have been working. Here is a close-up of the region where we see that a plateau is completely covered by forest, the first thing that caught our attention in the region. And if we can go back to 1967, we see that this forest is not recent, but was there before the larger occupation in the region that started around the 1960s, when they started to do selective logging of the region. Anyway, then the hypothesis we have to test is that Araucaria was originally restricted to the river valleys, starting around 4,000 years ago, when the Botavera cave records show a major increase in precipitation, and most of the paleovegetation records show the expansion of forests. We've seen that these climatic factors initially led to the expansion uphill of these forests, but natural conditions, but limited to the south-facing slopes. And in some areas, it's very likely that man expanded these forests beyond this potential natural distribution, starting around 1,000 years before the present. Okay. To test this hypothesis, we are doing transects of test pits of soil depth profiles, and what we are using is carbon isotopes that can tell apart very well grassland from Araucaria forest, as the work of Damin et al. has shown very well. Like in the region, grassland soils have a mean delta 13 of around minus 12.7, where Araucaria forest floor has been mean values of minus 28.8%, which allow us to differentiate in the profiles the changes in vegetation. So to end up, what we are testing, we have a control area south of the city of Lages in the state of Santa Catarina, where we know there is no archaeology at all, where we have the natural distribution. And we have the other area in Campobello. This is just 2 examples where we have 4 different transects in control and archaeological areas. And obviously what we expect to see is that in the close to the rivers, in the south-facing slopes, we would like to see across the profile a C3 signature, forest all the way. Then in the slopes, around 4,000 years before the presence, we would like to see a change from grasslands to forest. And finally, obviously in the north-facing slopes and the plateaus, we want to see grasslands continually. And the more important thing is that in areas where we have archaeology in the plateau, we want to see around 1,000 years before the presence this change from savannas to Araucaria forest. Thank you very much. I hope in the next pages I can present the results.
Which was the mechanism used by men to enlarge the forest? The fires, cutting trees?
This is a very interesting question. Today, the Sadanje people that live in the area do not live in reservations, do not propagate the forest. We know that Araucaria reacts very well to recently burned areas, and as we can see in most of the paleoecological records, there are peaks in charcoals before the expansion of the Araucaria forest, so it's very likely that the use of fire and the direct planting of the trees have taken place.
I would like to present you like a new DFG project that it's a collaboration between the Quaternary Geology Group at the University of Cologne, where my colleagues Bernd Wagner and Martin Mellis are located, as well as I, and between Jens Hölfort and Rich Pankost from the geochemistry unit, organic geochemistry unit at the University of Bristol. And we are actually aiming to record the vegetation during the early Pleistocene of the Lake Ohrid catchment. And it's like a period of time that unfortunately there are not so many records like from this region. So we are kind of like trying to do. Thank you. We are trying to investigate like in a period of time that we are pretty much left alone in this area. I would like to mention that the project is also located like in a bigger project, Skopsko project. And the pollen data from Lake Ohrid was published for the last 500,000 years by a bigger group of palynologists that is coordinated by Laura Sadori, which she's at the University of Sapienza in Rome. And my colleagues here, fellow palynologists Bertini, Cooley, and Donders, they help with some of the sampling processing because we are sharing a lot of samples for these skeleton diagrams. So let me go directly to the study area. In case you're not familiar with the region, we are in the southwestern Balkans, and Lake Ohrid catchment comprises also Lake Prespa catchment, which is like 150 meters higher. So Lake Ohrid, it's located at 690 meters above sea level. And the catchments are surrounded by mountain peaks above 2,000 meters. And they are like at a mid-altitude. There were several records published for the last glacial cycle from the two lakes, but the new ICDP core that was retrieved in 2013 reaches back to almost 1.3 million years. So this is a really long record that we have to deal with. A little bit about the climate of the region nowadays. You can see here the mean annual precipitation and temperatures. There is a strong gradient between the east and the west and the east coast, as you can see, like in precipitation. And there is a threefold decrease of precipitation between the west and the east coast. The climate of the region is transitional, sub-Mediterranean, we have mean values of temperature around 10 degrees Celsius. And there is also like a mean precipitation values between 700 millimeters per year in the lowlands, going up to 1,000 like at higher elevations. This is actually the core, the deep core. As you can see, it's a composite core. The retrieval rate, it's really almost 99%. My study interval is this red square. And I'm actually studying the onset of the lacustrine sedimentation. So it's between 450 and 400 meters. The first half of the core has already been extensively analyzed with different proxies, and there has been a special issue in biogeosciences covering the last 637,000 years. And we are now collectively working for the other half of the 570 meters. Here you can see the published arboreal curve from the Ohrid record plotted against age. And there are some orbital parameters here, and of course the benthic stack and the Tenagi-Philippon record, that it's another continuous lacustrine record from the region. This is the study interval once more, and what I would like to point out here is that we are in the 41 kiloyear world, so we are having like much shorter duration of glacial interglacials, and that's something that we would like to investigate, how the trees react in this period. So we would first like to find out how biodiverse was the catchment, and secondly, how the plant ecosystems within the catchment responded to climate variability in an obliquity-dominated world. In a recent review by Magri et al., there was a number of, I think, 100 records across the Mediterranean summarized for like paleobotanical records, and as you might be able to notice here, there's a big gap. Besides the Tenagi-Philippon record that I mentioned before, this is Lake Ohrid here, that was, of course, partly not included in the analysis because not all of the data are published yet. And there's also a site eastwards that it's not really Europe, but I mean it's a marine site in the neighborhood, so we can use it as reference. What you can see in this diagram, it's plotted against age once more. Here it's the time interval. And you have a summary of the pollen sites from southern Italy and Greece. I think it's more than obvious that there is a gap here. And from Italy, there is a site near Crotone that it was studied, and it's quite interesting that this site has, it's dominated by pines, along with quercus and tsuga, that they reach percentages up to 30%. Yes, this is what we intend to do with our record to fill this gap. And going eastwards, you can see the Tenagi-Philippon record. This site, it's located near the coast. It's a lowland site, so only 40 meters above sea level. And it's mostly dominated by oaks, but there are several other like relic species recorded in the record, but usually with percents below 2. The marine site here has, it's dominated by Taxodium, and some of the other relic species, tree species, are also with very low percentages. So this is the first results that I was actually counting till last Friday. So I was just putting this diagram together on my way here. And on the right-hand side, it's plotted against depth. I have added the time scale. The age on the left, and there are also the marine isotope stages that they're independent from my pollen zones, the pollen assemblage zones. And on the right-hand side, you can see the summary diagram of the trees versus the herbs, and it's pretty obvious, I think, that during the entire interval of the 200, almost 1,000 years that I'm studying, we dealt with a really forested area in the region. The other thing that I would like to draw your attention to is the curve of the pines, and it has been really heatedly discussed in our polling group what exactly is the role of pines and if it's overrepresented. In my case, there is dominance of pines during glacial times, and it's increasing towards the top of the interval. So having a look now from the region, here once more it's like plotted against age, the time interval, and here are the areas that they're neighboring to our, so southern Italy, Greece, Tenaghi-Philippon, and the marine side, and those are the species that I would like to briefly mention. So we see that in southern Italy, tsuga was reaching percentages of almost 30%, and this is almost the case, it's similar in Ohrid. So we have percentages around 25. In contrast to sites eastwards that they do not exceed 5%. Elcomia is also present in our record, but with very low percentages, and it's discontinuous. Parotia, the same. Liquidambar, the same. And Taxodium, there are some single grain occurrences. Sciadopitis, we didn't encounter. Cataya was also below 3 or 4%. And Cedrus, in our case, similar to Italy, it reaches percentages above 20. We didn't encounter Engelhardia, and Caria was an important component of the vegetation at Ohrid, like in southern Italy, reaching almost 12 or 13%. Pterocaria was like neighboring sites around 5%. And Zelkova also with values below 5%. So summarizing this together, this is what we have at Lake Ohrid. You can see cedars were really an important component of the vegetation, and they were reaching almost 40% in MIS 37. Then we had, I would call it like a separation of the record, like the upper half shows high percentages of conifers like tsugas, and there's also like more of caria pollen and pterocaria, in contrast to the lower half. And most of the species that we have here are, I mean, in percentages, like the Parotia and Elcomia, they're really negligible and they're just discontinuous in the record. So, in order to put this together, I would like to mention that we were expecting that the site, because of the location on the western flanks of the Balkans, it's going to host a higher biodiversity and higher percentages of trees in comparison to Tenaghi-Philippon. It's also known from the area, the record of Ioannina, that it's a refugial site, so Western Balkans really hosted forests, and we also see that the landscape was rather forested. There are also parallel analyses that my colleague Jens Hölfert is doing with lipid biomarkers, and we concluded that we had a rather stable soil pool. We will try to combine our two data sets within the following weeks, and within the next year, we are planning to. I'm planning to produce like a high-resolution pollen record. This was only like at a temporal resolution of 2,000 years. We are planning to go down to a centennial resolution. So, in case you're interested to find more about the biomarkers, you can visit on Saturday, the poster 64. And I would like to thank you for your attention.
The title of my presentation is Spatio-Temporal Dynamics of Bacterial Communities in Response to Marine Transgressions and Regressions Occurred Since Late Pleistocene. So why do we need to explore bacterial community response? Because bacteria is first evolved on Earth. And this picture shows stromatolites, the oldest fossils of cyanobacteria. And these cyanobacteria transform the cold carbon dioxide-filled Earth into an atmosphere suitable for life called Great Oxygenation Event. And since then, they started controlling the biogeochemical cycles and thereby the climate. So sea level fluctuation is an important feature of climatic changes. And studying the response of bacterial communities from the past climatic changes is important in predicting the future carbon dioxide and oxygen levels. This study is a part of a major research project, and the aim of which was to demonstrate and validate the ability of bacterial communities as high precision sea level indicators. And the objective of the present study is to reconstruct past bacterial diversity from ancient DNA and to investigate bacterial community response to marine transgressions and regressions. So the sampling location we chose was a beach ridge. Since beach ridges are geomorphological indicators of paleo sea levels, and using geomorphology map, we located a paleo beach ridge at Veitekaran Iruppu, and we named it as VKI, which is situated 2.6 kilometers inland from the present shore and located on Kaveri Delta, Nagapattinam district, Tamil Nadu, southeast coast of India. The elevation with respect to mean sea level of the beach ridge was 7.2 meter, and a sediment core of 25 meter depth was excavated through calyx core drilling. Micropaleontological and geochronological analysis were also carried out. So how do we acquire past bacterial community data? Ancient DNA was isolated by CTAB method from the sediment layers, and V3 hypervariable region of 16S rDNA of bacterial genome was selectively amplified using specific primers, and the amplicons were then run on NextSeq 500 Illumina sequencing platform. The raw data were pre-processed and analyzed using KIME and R software. So these are the results of OSL dating and micropaleontology. OSL dating of the sediment showed Holocene epoch till a depth of 11.9 meter, and from 12 to 25 meter depth it was Pleistocene epoch. So moving on to foraminiferal data. At 21 meter depth there were only few foraminifera, and which corresponded with last interglacial transgression, whereas from 5.6 to 10.9 meter, molluscous shells, abundant and diverse population of foraminifera were observed, which corresponded with mid-Holocene transgression. So based on OSL dating and micropaleontological studies, we chose 4 samples for bacterial community reconstruction, and one from bottom, one from the top, and the 2 other samples were based on the foraminiferal evidences. The bottommost sample, VKI, is named after the coring location, and the number was based on the core tube. And VKI-14 was chosen at a depth of 24.5 meter, which was 146,000 years old, late Pleistocene epoch. And VKI-12 was chosen at a depth of 21 meter, which was 104,000 years old. And then VKI-5 at a depth of 7.2 meter, which was 6,000 years old, mid-Holocene epoch. And VKI-2 was chosen at a depth of 2.8 meters, which was late Holocene, 3,000 years before. So VKI-12 and VKI-5, they showed marine proxies, that is foraminifera, and the bacterial population were affected by sea level rise, whereas VKI-14 and VKI-2 did not show any marine proxies. And VKI-14 was considered as native bacterial population. VKI-2 was the youngest sediment and considered as final community, and considered having niche conditions similar to VKI-14. So the results of DNA, least DNA yield was observed in VKI-5. In spite of that, high 16 rDNA sequences were observed in VKI-5 when compared to all the samples. Moving to diversity analysis, the rarefaction curve of VKI-2, VKI-14 and VKI-12 became flatter to the right and had almost reached plateau. And this indicates good sampling and sequencing. And more intensive sampling could yield only few additional species, whereas VKI-5 had the steepest rarefaction curve, and more intensive sampling and could yield additional species. And diversity indices, 70% of the estimated diversity were observed in VKI-2, 12 and 14, whereas only 57% of the estimated diversity could be recovered. In spite of that, highest species diversity was observed in VKI-5, and species richness and species evenness were also highest in VKI-5, calculated by Shannon and Simpson index. So moving to phylum level distribution of bacteria, VKI-14 showed 16 phyla totally. Moving on to VKI-12, 7 phyla disappeared in VKI-12 and 5 new phyla appeared. Moving on to VKI-5, it had the highest phyla and total of 37, and 20 new phyla appeared in VKI-5 and 6 phyla which disappeared in VKI-12 reappeared in VKI-5. Moving on to VKI-2, no exclusive phyla were observed, but 17 phyla of VKI-5 disappeared in VKI-2. So these are the commonly shared phyla among all the 4 samples. WS5 was the only exclusive phyla of VK14. These are the 3 phyla exclusive of VK12. And these are the list of 14 exclusive phyla of VK5. These are the list of 5 phyla which are commonly shared between VK14, VK5 and VK2. And TM7 was the only phyla shared between VK5 and VK12. So these pie charts show the shift in bacterial community structure from when moving from one sediment, from the bottom sediment to the top. And Proteobacteria, Bacteroidetes, Firmicutes, Actinobacteria, Vericomicrobia, Chloroflexi were the abundant phyla, out of which Proteobacteria was the dominating phyla. So to calculate the similarities between bacterial communities, Sorensen index was used, and VKI14 showed higher similarity with VKI2. Coming to habitat annotation, aerobic bacteria and terrestrial bacteria were high in VKI14 and VKI2, whereas anaerobic population was dominating in VKI5. Moving to bacterial community dynamics in VKI samples, 35% of the bacterial communities were able to move from VK14 to VK12, and 40% of the bacterial communities were able to move from VK14 to VK5, and 57% of bacterial communities were able to move from VK5 to VK2, and 10% from VK14 to VK2, and 5% from VK12 to VK2, and 28% were unable to adapt to the changing ecological conditions, whereas 25% of the population were able to adapt and move from VK14 to the top samples. So with this study, yes, species do move, adapt, and die. To summarize, during last interglacial transgression, bacterial diversity decreased, and during mid-Holocene transgression, bacterial diversity increased. And introduction of exotic species and adaptation of native species to the changing environment increased the bacterial diversity. Although marine transgression regressed, resulted in introduction of exotic species, 64% of the species were unable to survive after regression. To conclude, bacterial DNA were able to sustain activity even after 146,000 years in spite of low metabolic rates, and bacterial diversity increased by marine transgression through introduction of exotic species. And past environmental conditions are reflected by sediment bacterial communities, thereby which bacterial diversity dynamics can be used for predicting past sea level changes. So the authors acknowledge Ministry of Earth Sciences, Government of India for funding this work, and pages for the Travels grant. Thank you for your attention.
Did you find any association between the sediment composition and
yes.
bacterial composition?
Yeah, that's what. In VKI5, I said the DNA yield was least. In spite of that, we were able to get high 16S rDNA sequences. Yes. Here, the DNA yield was least because of the the strata of the sample sediment, which was compact clay. Generally, clay has the high ability to, have high affinity towards DNA, and therefore the least DNA recovery was observed in VKI5. Whereas when compared to other sediments, VKI5, in spite of that, it has a high 16S, species diversity observed.
You mentioned something about the anaerobic bacterial population in VKI5.
Yes.
So do you correlate that with the sediment nature or the soil composition?
Yeah, here the transgression was around 3,000 years old. So thereby the anaerobic condition was observed. Whereas in case of VK14, regression condition was observed, so that the terrestrial conditions were present that time, and aerobic population was dominating there in the native bacterial community sediment,
okay.
the bottom sediment.
Thank you.